This is the review for Unit 1, Food for Life. You can access this review at srhsbio.wikispaces.com and on YouTube channel LaFell Labs. First question asks us to convert a cell, which is 5 micrometers in diameter, into millimeters. So the first thing we should do, we're trying to convert 5 micrometers. So we're going to go ahead and write 5 micrometers. And underneath that we have 1. It's a placeholder. Now I want to change micrometers into millimeters. So if I have micrometers on top, that means that I need micrometers on bottom. This way they cancel out. Micrometers on top of micrometers cancels out to 1. Once those unit cancels, I need to put millimeters on top so that way my answer will be in millimeters. Now that I've set up my conversion, I just need to add the numbers. If we look at our conversion factor, 1 is with millimeter and 1,000 is with micrometers. So let's keep them together. This means that we have 1 millimeter on top and 1,000 micrometers on bottom. So to solve this is the same thing as moving the decimal over 3 times. We're just going to do 5 divided by 1,000. So it's 0.005 millimeters. Then we can ask ourselves if this makes sense. A micrometer is smaller than a millimeter. So if I have 5 micrometers, I'm going to have fewer millimeters because millimeter is bigger than micrometer. Next question. A group of students wish to determine which brand of fertilizer is best for growing daisies. They collected the data below. So they have 4 experimental groups. They used No Fertilizer, Miracle-Gro, New-Gro, and Supergreen, and they measured the growth in centimeters. First question asks us, what is the independent variable? The independent variable is we are trying to see the effect of. It is what I vary for each trial. So independent variable and I vary both have IV in them. So it is what you are intentionally changing for each trial. Here we have 4 different trial groups, and we see that the brand of fertilizer is different for each of them. So we are trying to see what effect fertilizer has on our growth in centimeters of our daisies. Next question asks for the dependent variable. The dependent variable is what we are trying to measure in the experiment. It is the effect that we are trying to see, and it depends on the independent variable. So in this case, the growth in centimeters depends on the type of fertilizer that we use. Therefore, that is our independent variable. Next question asks us, what is the control? So the control is a comparison. It's how we know that the independent variable did anything at all. The trial without the effect of the independent variable. So our independent variable is the brand of fertilizer. Therefore, the experimental group without any fertilizer being used is going to be my control, which is group A. This is useful because if I don't have a control, I don't know how much better the fertilizer makes the daisies grow. So I could have gotten that Miracle-Gro only had 10 centimeters in growth, in which case, if I was trying to grow daisies, I wouldn't go waste money on Miracle-Gro because no fertilizer works just as well as Miracle-Gro would have. We can see here that New-Gro only had a little bit more of growth, so I probably wouldn't want to waste money on that one, even if it was the cheapest fertilizer to buy, because it doesn't really do much for my growth. And what are some constants? So in an experiment, you want to change your independent variable and measure your dependent variable. So those are the only things that should change. Everything else needs to be constant. So we need to use the same species of daisy for group A, B, C, and D, the same type of soil, the same amount of water, same amount of sunlight, same amount of carbon dioxide. All of these things need to be kept constant. Otherwise, if let's say I exposed each of my experimental groups to a different amount of sunlight, how do I know that the reason Miracle-Gro grew 12 centimeters or Super Green grew 15 centimeters? How am I certain that it's just because of the fertilizer? If it's a different amount of sunlight, maybe that is why they grew differently. Next question asks us to place the electrons. So, in a carbon atom, the first energy level can hold up to 2 electrons. There's 1 electron and 2 electrons. The second energy level can hold up to 8. Now, carbon has a total of 6 electrons. 2 are in the first energy level, which means that my second energy level must have 4. So here are my 4 electrons in the second energy level. Now for carbon, this is a really big deal because this allows carbons to form very complex structures. So carbon has 4 valence electrons. Those are the ones in the outer energy level. But it wants to have a total of 8 to be stable. We call that a full octet. To get those extra 4 electrons, carbon can form up to 4 bonds with other atoms. And each chemical bond represents the sharing of 2 electrons. So by sharing 4 bonds, by making 4 chemical bonds, I have a total of 8 shared electrons. Next question asks us to explain the formation of an ionic bond. So in this picture, I have a sodium atom and a chlorine atom. And these guys have, in the outermost energy level, sodium's got 1 valence electron, and chlorine has 7. Now, if I were to transfer 1 electron from sodium over to chlorine, sodium lost an electron, so its charge would be +1, and chlorine gained an electron, so its charge would be -1. We call the positively charged atom the cation, and the negatively charged atom the anion. These guys have opposite charges, and opposite charges attract, and this is what forms the ionic bond. How is this different from a covalent bond? So an example of an ionic bond. Let's say that Mason has a set of markers, but he's missing the yellow one. So Paige gives him the yellow marker. One atom gave the marker, aka the electron, to another atom. This would be an ionic bond. 1 electron or more is transferred to another atom. A covalent bond would be like Emily has all of the markers but green and red, and Will has all of the markers but blue and yellow, so they share markers so they each have a full set some of the time. In covalent bonds, atoms share electrons so that they have a full octet at least some of the time. So that's what we see with carbon. Carbon can make up to 4 bonds, and it shares electrons in those bonds so it gets a full octet at least some of the time. The next question is about acids and bases. First, it asks us to compare the levels of hydronium and hydroxide in acids and bases. Those square brackets mean concentration of. So the question is asking, compare the levels of the concentration of hydronium and the concentration of hydroxide in acids and bases. So here I have 2 different solutions, and the red dots represent hydronium and the blue dots represent hydroxide. And we can see in the first solution that hydronium has a greater concentration than hydroxide. This means that the first solution is an acid. Also, H plus and H3O plus, they are the same thing. They are both hydronium. In the other picture, we see that hydronium concentration is less than the concentration of hydroxide, which means that this picture shows us a base. The pH levels for something that is neutral, that is where hydronium concentration is equal to hydroxide concentration, the pH will be 7. Things that range from 0 to 7 are going to be acidic solutions. This is where hydronium is greater than hydroxide. And things greater to 7 to 14 represent bases. And these have a hydronium concentration less than the hydroxide concentration. Next question asks us, what is the function of a buffer? A buffer is something that reacts with acids and bases to prevent sudden changes in pH level. So, let's say that we have a lake, and the lake is at pH 7, and living inside the lake is a nice, happy, little single-celled organism. One day, acid rain cloud comes along and rains acid rain down on top of the lake. The lake becomes acidified. Its pH becomes lower, meaning that there's more hydronium concentration, and the pH goes down. This is bad news for the single-celled organism that lives inside the lake. If the lake were protected by a buffer, the acid rain would not have changed the pH of the lake. This cell was not able to maintain homeostasis with the sudden pH change, so it died. Homeostasis is just the narrow range of conditions that an organism needs in order to stay living. If those conditions suddenly change, such as with our sudden pH change, the organism cannot maintain homeostasis and it will die. Blood is also buffered so that whenever you eat something that is acidic or drink an acidic drink, the pH won't change. Next question asks you to prove and illustrate each statement. So the first statement is that water is a polar molecule. So here we have a water molecule. It is 2 hydrogens and 1 oxygen. Inside of the oxygen atom, it has 8 protons. Inside of each hydrogen atom, there's 1 proton. So each proton has a +1 charge, so that means the oxygen is overall +8, and the hydrogen is overall +1. Now, if you're an electron, you're going to be more strongly attracted to the oxygen because it has a +8 charge. What that means is that the electrons tend to spend most of their time near the oxygen. Because they don't really hang out too much around the hydrogen end of water, that end becomes slightly positive. Since the electrons are spending so much time around the oxygen end becomes slightly negative. So this is why water is a polar molecule. You have one side that is slightly positive and one side that is slightly negative. And that's just due to where the electrons are spending their time based on the number of protons in each atom. Next question is about how water is able to form hydrogen bonds. So here I have 2 water molecules, and when they get close to each other, they form an attraction, and that attraction is the hydrogen bond. Each hydrogen is slightly positive, while each oxygen is slightly negative. The attraction between the two is a hydrogen bond. They're oppositely charged and opposites attract. Hydrogen bonds are being constantly broken and reformed as the water molecules are moving about. So this hydrogen bond that we just formed, it might disappear the next second and a different bond might reappear the second after. They're very fluid and they're broken and reformed as the water molecules move. The next statement is that water is able to dissolve many things. So water is a polar molecule, we already explained that, and its charged ends are able to attract to other things that also have a charge. This allows water to rip apart charged particles, such as the salt crystal that's going to be in this video. One way that we can summarize water's ability to dissolve things is with the phrase like dissolves like. In other words, something with charges will dissolve other things with charges. Something without charges will only dissolve things that are not charged. So this is why oil and water don't mix. Water is polar, oil is nonpolar. As in water has charges, oil does not. Therefore, they do not mix.
Sodium chloride crystals are held together by attractive forces between the positively charged sodium and negatively charged chloride ions. When a crystal of sodium chloride is placed into water, the hydrogen ends of polar water molecules attract the negatively charged chloride ions and gradually surround them. Likewise, the oxygen ends of water molecules are attracted to and surround the positively charged sodium ions. The hydrated ions drift away into the solution, allowing new water molecules to surround newly exposed ions. Gradually, the entire crystal dissociates into solution.
Next statement says that water has a very high surface tension. So water is able to form hydrogen bonds. Therefore, water is more attracted to itself than it is to air. This leads to surface tension. So here in this picture, we see the water molecules, and we see that they're forming lots of attractions with each other, but no attractions with the air. So this leads to a water droplet with very high surface tension. Some bugs even make use of this to walk on water. Number 8 asks us to draw the structure of a polysaccharide. So a polysaccharide is many monosaccharides or single sugars linked together. So here I've got a bunch of sugars, they're linked together. That makes this a polysaccharide. A disaccharide is where we have 2 sugars linked together, and a polysaccharide is many sugars linked together. These are also sometimes called starches. Next question asks us to draw the structure of DNA. So the sugars and the phosphates, they alternate to form the backbone. The nucleotides line up in the middle, and they are held together by hydrogen bonds. A and T pair up, and G and C pair up. Next question asks us to draw the structure of a protein. So a protein is going to be many amino acids linked together by peptide bonds. Therefore, we call a polypeptide chain a protein. So here I have many amino acids that have been linked together by peptide bonds, and this is a protein. Draw the structure of a fat. So 3 fatty acids held together by a glycerol will give us a fat. The fatty acids can be all the same, or they can be different from each other, and they might be saturated or unsaturated fatty acids. So here I have a glycerol joining together 3 different fatty acids. Fatty acids may be saturated or unsaturated. The difference between the two is that in a saturated fat, all of the fatty acids have single bonds. Every single carbon, hydrogen, it's all single bonds. In an unsaturated fat, here we have this double bond. Unsaturated fats have no kinks in this tail that forms the fatty acid. Therefore, they're able to pack in very closely together. So these are going to be solids at room temperatures. So that's going to be things like butter. The unsaturated fatty acids have these kinks in them, and that prevents the fatty acids from packing in together. So these are going to be liquids at room temps, and this is things like olive oil. How do carbohydrates form bonds? So the way that they form bonds is through something called dehydration synthesis. If I have 2 monosaccharides, I can join them together into a disaccharide, losing water in the process. I can repeat this several times and form a polysaccharide. Here I've got a glucose and a fructose. They're going to form a bond there. We lose water, and I've now formed the disaccharide sucrose. Next question is to complete a table about DNA and RNA. So DNA is deoxyribonucleic acids. That's where DNA comes from. And RNA is ribonucleic acid. The sugar for DNA is going to be deoxyribose, and for RNA it's going to be ribose. So they're both ribose sugars, it's just that DNA is deoxyribose. What that means is that ribose is missing an oxygen. Both are a polymer of nucleotides. The nucleotide is the monomer. Many of them together makes a polymer. The bases found in DNA are A, T, G, and C. Adenine, thymine, guanine, cytosine. In RNA, we have AUGC, adenine, uracil, guanine, and cytosine. DNA is double-stranded, whereas RNA is single-stranded, and they both contain a chemical code or message. The last question has a table with information for you to fill in about different macromolecules. The first macromolecules is going to be carbohydrates. Carbohydrates are the main energy source for living things, and they also have structural purposes as well. So important examples are glucose, which is blood sugar, and sucrose, which is table sugar. Notice that these both end with -ose. That's usually a good hint that something is a sugar. Another example is chitin, and this is a starch. It is the exoskeleton of things like grasshoppers, insects, and horseshoe crabs. The monomer for a carbohydrate is a monosaccharide. So that's just a single sugar like glucose. Polymer is going to be either a disaccharide or a polysaccharide. So here we see we have a glucose and a fructose that have joined together, and that makes sucrose, which is table sugar. A polysaccharide would be a starch, and in this particular example, we see that we have several individual glucose molecules held together by several bonds, and that is a starch. Glucose isn't the only monosaccharide. Any single sugar, fructose, any of those things are going to be monosaccharides as well. Lipids. Lipids are used for long-term energy storage. These are fats. They are also used in cell membranes. Steroids and hormones are another important group of lipids. These are involved with a lot of regulation functions in your body. Important examples, phospholipids make up the cell membrane. So each and every single one of the cells in your body has phospholipids as the cell membrane. And these phospholipids, one end of them is hydrophilic, and another end is hydrophobic. So if you drop these into water, they actually arrange themselves into this bilipid membrane where the hydrophobic parts are protected by the hydrophilic parts. The monomer, so for a lipid, it's going to be a glycerol and 3 fatty acids. For a phospholipid, we're going to have a glycerol, a phosphate, and 2 fatty acids. So that phosphate is the part that is hydrophilic, whereas the fatty acids are the parts that are hydrophobic. A polymer could be the fat formed from several lipids together, or a phospholipid bilayer. Proteins. Proteins are used for all kinds of stuff within the cell. They control rate of reactions, they can regulate cell processes, they can form cell structures, they're also involved in transporting things into and out of the cell. Proteins are just used for all kinds of stuff within the cell. An important example would be lactase. So this is the protein that is responsible for breaking apart milk sugar or lactose, breaking it down during digestion. So people who have not enough lactase or their lactase is not formed correctly, they're going to be unable to break down lactose and they're going to be lactose intolerant. Notice that this ends with ase. A lot of proteins end with ase, so that's a good hint that something is a protein. The monomer for a protein is an amino acid. Now there's 20 different amino acids that occur in nature. The amino group and the carboxyl group are going to be the same on every single amino acid. These are what allow amino acids to link together to form a protein. Now the R group, which is here in purple, the R group varies. Depending on what the R group is, tells me the type of amino acid that it is. The polymer is a polypeptide chain. It is many amino acids linked together by polypeptide bonds. So here I have an aniline and a serine. The only difference between these two is that purple R group, and whenever I link those together, I form a peptide bond. Many of these different amino acids linked together would be a polypeptide chain. Vitamins and minerals. They help various chemical reactions occur in the body and in cells. And important examples would be vitamin C for wound healing, vitamin D for bone formation, vitamin K for blood clotting. Calcium is an important mineral that makes up bones, so your bones are actually made of calcium phosphate. And iron is found in hemoglobin. This is the protein in blood that allows blood to carry oxygen to all of the different parts of your body. Nucleic acids. So nucleic acids store and transmit hereditary information, and they also direct protein synthesis. Important examples are going to be DNA and RNA. The monomer for both DNA and RNA is called a nucleotide. It consists of a phosphate, a sugar, and a base. Depending on whether you have DNA or RNA, that sugar is going to either be deoxyribose for DNA or ribose for RNA. And the base can be adenine, thymine, guanine, and cytosine for DNA, or adenine, uracil, guanine, and cytosine for RNA. The polymer of many nucleotides is going to be a nucleic acid. Double-stranded nucleic acid is DNA, and single-stranded nucleic acid is RNA. Some tips for taking your test. Complete your review. If you have questions or you don't understand something, circle those questions and then ask about them with your teacher. Make flashcards for tricky topics only. So making them for every single topic is going to take you forever and it's going to be a waste of time. Focus on things that you know that you are struggling to understand, and then you have specific flashcards for those topics without wasting your time on the stuff that you already know. Look over your previous quizzes. So check which questions. Look over previous quizzes. Check which questions you got correct and find the answers to the ones you got wrong. The night before your test, take about 20 to 30 minutes and go over your review. Get a good night's sleep, and in the morning, eat breakfast. Again, you can access this review at srhsbio.wikispaces.com or on YouTube channel LaFell Labs.